Hi, Gen Bio. Dr. Fry here. This week we are going to be discussing eukaryotic chromosomes. So basically we're going to work our way through the cell cycle and take a particular look at the stages of nuclear division. Okay, so just by way of an introduction, the cell cycle includes interphase, which is where the cell is going to be spending most of its time. This is when the cell is undergoing its normal functions, it's growing, and it will also be duplicating chromosomes as well as the organelles within the cell to prepare for nuclear division. Okay, so interphase is where it spends most of its time. Nuclear division is going to be what we're going to take a more in-depth look at during the course of the lab this week. And then cytokinesis is following nuclear division. That is the division of the cytoplasm between daughter cells. So it happens after the nucleus divides, after the chromosomes are partitioned, but it's the partitioning of the rest of the material within the cell. Okay, so interphase, it consists of three different stages: G1, S, and G2. Okay, so again, this is when the cell is undergoing a lot of normal function. So if it's, say, a neuron, it's going to be sending signals. If it's a muscle cell, it's going to be working with other muscle cells to contract your muscles and do work, and so on and so forth. So G1 is a normal functioning growth stage of interphase. The S stage of interphase is particularly noteworthy for us because we want to take a special note that this is where there's replication of the chromosomes. Okay, so within our nucleus, we have all the genetic material for that cell encoded in chromosomes. And DNA synthesis and chromosome duplication takes place during the S stage of interphase. Okay, and so when a chromosome replicates all that genetic material, when all that is duplicated, then we have basically what we call its sister chromatid. Okay, so I have in beads here a model for our chromosomes. Okay, so this is a chromosome. During S stage of interphase, all of this genetic material is going to be duplicated, and then it will have a sister chromatid. Okay, and that's again exactly the same genetic material that has been replicated in preparation for division, so that the one cell can produce two daughter offspring cells. Okay, and then G2 phase is also part of interphase just prior to nuclear division. Okay, so nuclear division, when I was a kid, there were only 4 stages of nuclear division: prophase, metaphase, anaphase, and telophase. Okay, now some sources will also include pro-metaphase as a 5th stage of nuclear division. Sometimes it's referred to simply as late prophase, and then prophase is called early prophase. Some sources will distinguish then early prophase as prophase, and late prophase as prometaphase. Okay, so this is noted in your lab manual as well. So just be familiar with the interchangeability of those two terms. Okay, so prophase, the first stage of nuclear division, is when, if you are looking under the microscope at a cell, you will now be able to see the chromosomes more distinctly. Whereas before, if you were looking at the cell, the nucleus would just be kind of a hazy gray color. It would not be particularly, you would not be able to notice the chromosomes as distinct structures, okay? Whereas now that they are condensed in prophase, now you can see them as much more darkly pigmented and more distinct structures. And then prometaphase is just distinguished from prophase in that now the nuclear membrane or the nuclear envelope that had contained all that material in the nucleus has started to break down, has started to be disrupted, it's fragmented, so that then the chromosomes, which are held within the nucleus, can migrate to the different sides of the cell in preparation for the division into two daughter cells. Okay, so prior to division though, there's a few more stages. Okay, so prophase, you can start to see the chromosomes condense. Prometaphase, you've got fragmentation, breaking up of the nuclear envelope. And then metaphase is when, if you're looking under the microscope, you can see that the chromosomes line up in the middle of that cell. And we call this the metaphase plate. Okay, you're gonna see the chromosomes form a pretty nice line right in the middle of the cell. And then anaphase follows, metaphase. This is when the chromosomes will be pulled to opposite ends of the cell. So the two sister chromatids that we had from duplication of the one chromosome, they had previously lined up in the center of the cell during metaphase. Now they are being pulled apart. Now they're being pulled apart to opposite ends of the cell. So these two sister chromatids will be pulled apart. One will go to this daughter cell, one will go to this daughter cell, and that movement towards the poles is what distinguishes anaphase from the other stages of nuclear division. Okay, so anaphase, the chromosomes, which had been sister chromatids, now move to the opposite sides. And I'll mention that they had been bound at the centromere. So they had been attached at a particular point in those sister chromatids. And that, in my little model, is right here, okay? They had been bound, and that's the centromere, the point at which they were bound, okay? All right, and then telophase is the last stage of nuclear division. And this is just when you get a reorganization of the two daughter cells. You can start to see the nucleus taking shape in each of the two now new cells, okay? And then after the stages of nuclear division, then you have partitioning, like I said, of the cytoplasm and all the other organelles that will be distributed to the two daughter cells, okay? Now this looks a little bit different if you're looking at an animal cell versus a plant cell. And this is, in animal cells, it's seen as a cleavage furrow. So you're gonna see like a little bit of, almost like a constricting, narrowing band appearance between the two daughter cells. So you had two cells, it's going to start to form the cell membrane. There's going to be a constriction formed between the two before they ultimately divide. And then in plants, you're going to start to see what's called a cell plate form, which will ultimately become the cell wall for the plant cells, okay, for the two daughter cells. All right, so I've used a couple terms that I just want to make sure we rehash so you know what, so we're all on the same page, okay? So when I say chromatid, that is referring to each strand of a previously duplicated chromosome. So where you had one chromosome, okay, with genetic information, all your genes on there, during S stage of interphase, all of that is duplicated, and then the two are called sister chromatids. That's duplicated genetic information, okay? And that's shown here in the picture as well. So you have one chromosome, all that's duplicated in preparation for nuclear division. So you now have sister chromatids, okay? And they're attached at that point called the centromere, right there, okay? Now, other terms that you will see in your lab manual, this are a part of the spindle apparatus. So the spindle apparatus, I've included a picture of it here, the bottommost picture. So there's a lot of different proteins that are involved in this thing called a spindle, okay? And these are seen in animal cells, and they are lots of proteins that work together to pull those sister chromatids apart and distribute them to the two daughter cells. Okay, so in the picture, if you look, the green stained structures, these are microtubules. They form like a star shape here, almost a star shape on either side at the poles of the one cell, of the one parent cell. And they are working to pull apart the sister chromatids with help from the kinetochore. Kinetochore is shown here in pink. These are structures which help, again, to pull apart the sister chromatids. The centrosome is a set of proteins here in the center of the aster. You cannot actually visualize them in this picture, but they also contain centrioles that are in the center of that aster and work together as part of the spindle to draw the sister chromatids to the sides, to the poles of the parent cell in preparation for those two daughter cells to be formed. Okay. Okay, let's move to our next slide. So there are two types of nuclear division that we're going to discuss. The first of which is mitosis. So this is one type of nuclear division that happens in almost all cells. So it's occurring in plant meristems, animal bone marrow, animal epithelium, lots of healthy growing areas. It does not directly give rise to production of the eggs and sperm, our gametes. That will be meiosis, which we'll talk about in just a minute. And if the cells are already differentiated and will not divide any further, then it may also not take place. But any healthy growing areas, those are going to have cells undergoing mitosis at various points. So this is a form of nuclear division that is producing two sets of genetic information that will ultimately become two daughter cells. And important feature is that you have a replication of each of the chromosomes, one division, so then you have the same exact number of chromosomes as you began from the parent cell. So each of the daughter cells, there will be two, has the exact same number of chromosomes as you had in the parent cell. Okay, and the 4 or 5 different stages of nuclear division all take place. Prophase, prometaphase, metaphase, anaphase, and telophase all take place one time. And as a result, you get two daughter cells with the exact same number of chromosomes as you had in the parent cell. All right, so let's take a look at some pictures that will look similar to the slides that you will be looking at in lab this week. So I've included these photos of each of the different phases of nuclear division. Now these are all plant cells. Okay, you're going to be looking at plant and animal cells in lab this week. But I'm pointing out in the first picture, this is an example here of prophase, and I will just distinguish it from this cell here, where you can see the nucleus and the nucleus and the nucleolus. And that would indicate that that cell is still an interphase. It has yet to undergo nuclear division, okay? When it does, and it starts to enter prophase, you're gonna start to see the chromosomes become a little bit more distinct, and you're gonna lose the shape of the nucleolus in the nucleus, okay? It's gonna look more like this. Late prophase or prometaphase, stage 2, is when you then lose that nuclear membrane. So you can kind of still see there's a structure containing the genetic material still existing in prophase. But in prometaphase, you really start to see more of those chromosomes bunch up. You can see them become condensed and more visible. And you do not any longer see that nuclear membrane holding that material, okay? So it has progressed, and this would be another example, to the point where you can't see that nuclear envelope anymore, okay? So let's move down to metaphase. Metaphase, I had said, is when you start to see the chromosomes line up at the metaphase plate in the center. And that's exactly what's happening in this picture here. Okay, you can see they're moving to the center. And then just after that, in anaphase, the sister chromatids are now their own individual chromosomes. They've been pulled apart. And you can see they're moving to the poles. So this is a very nice demonstration where you can see all of these sister chromatids are being pulled to this side, and these guys are being pulled to this side in preparation for telophase, where you can start to see, now this is a plant cell, so you can start to see there's a cell plate forming right here. I don't know if you can see that, but there's a little division forming, okay? And now you can start to see the reorganization of the two daughter nuclei, okay? All right, let's move on. I have a few more pictures in the next slide. Just showing you again another example of prophase and prometaphase. Here, they're described as early and late prophase, but again, remember that those are the same, prophase or early prophase, and prometaphase or late prophase. And again, the distinction is you can still kind of see that there's a fragmentation, but there's still a nuclear membrane that's bound, that all of this genetic material is still bound by. Whereas in late prophase, you can see that that has been broken down enough such that the chromosomes have started to move and are no longer contained by that nuclear membrane. Okay, and now let's move to metaphase, where again you can see the lining up of the chromosomes at the metaphase plate. And in this picture, now this is an animal cell. Okay, this was a plant cell as well. You can see by the cell wall. This is an animal cell, and you can see in this picture you've got a faint spindle observable here. Okay, so the center of that spindle are the centrioles, and you can see the microtubules here forming that aster. And again, those are helping to move the sister chromatids apart. They'll shorten and pull those sister chromatids to either ends of this parent cell. Okay, so let's move down to anaphase, where you can again see that happening, the movement. You can start to see there's two structures here forming. Now these sister chromatids are their own individual chromosomes moving to either of what will become the daughter cells. Okay. And again, you've got telophase. You can start to see a cell plate forming here, which will become the cell wall in plants. Okay. All right. So let's move to our next slide. We talked about mitosis, which is one type of nuclear division. The second type that we have to talk about is meiosis. Meiosis is the type of nuclear division which will give rise to our sex cells, our egg and sperm, also called the gametes, okay? So this is going to take place prior to sexual reproduction. And the big important point here is that whereas in mitosis, we had one round of chromosome duplication and one round of division, nuclear division, here in meiosis, we have one round of chromosome duplication, just like mitosis, but two rounds of nuclear division. So you're going to go through those phases: prophase, metaphase, anaphase, and telophase twice. Okay, so prophase through telophase 1, this is the first round of meiosis, and it's going to separate what we call homologous chromosomes. Okay, so let me tell you a little bit about homologous chromosomes. In humans, we have 46 chromosomes, or sometimes worded 23 pair of homologous chromosomes. Okay, so 23 pair of chromosomes, and homologous chromosomes are those that look similar. So you have 23 pair, 22 of those pair are somatic chromosomes, and then the 23rd pair are the X and the Y chromosomes, okay? So for the 22, they look identical to their homologue. So this is one chromosome. This is a homologous chromosome. This is chromosome 1, and this is chromosome 1. But you got, when you were just being fertilized, you got one of these sets from your mother. One's maternal and one is from your father. The other is paternal. So you inherited genetic information from your mother and your father. And this genetic information, while the actual trait that it may encode for on that chromosome may be different, the information is the same, is encoded for at the same location on that same chromosome. So for example, on chromosome 1, or I'm just making this up, which I don't know which chromosome it's encoded for, but on chromosome 1, you might have a gene that encodes for hair color. You may have inherited a different form of that gene, a different allele from your mother than you did from your father, but the trait would be encoded for at the same location on the same number chromosome. So we call that location a loci, the location on the chromosome where you have a specific gene, okay? So that gene encodes for, say, hair color or eye color, whatever it be. You might get different forms of it from your mother and your father, but you have one chromosome and its homologue inherited from each of your parents, okay? So I hope that makes sense. That's what homologous chromosomes are. So a distinct feature of meiosis is that when you have the chromosomes line up at the metaphase plate, in metaphase 1, it's actually the homologous chromosomes that line up next to each other. So they have each undergone division already and they have each undergone replication already. So I'm going to enlarge this picture just a bit to show you that. In the picture, you can see that the parent cell, for this example, had 6 different chromosomes. Okay, and there are 3 pairs of chromosomes. So there are 3 different sets of homologous chromosomes. So you can see that this one is shorter. This is its homologue. This one is longer. This would probably be its homologue. So they all line up next to each other. They pair up next to each other. They've each been replicated during S phase of interphase. So now you can see that this one red chromosome duplicated its genetic material. So now there are 2 sister chromatids that are both red. And the blue one also replicated its genetic material. And so now you can see there's 2 sister chromatids. But all 4 of those, all 4 will pair up next to each other. And this is called synapsis. Okay, so they'll line up next to each other. And they're, of course, with their homologous chromosome and the sister chromatid. Okay, so these guys are lined up next to each other during metaphase 1. Another thing I'll point out here is that you can have, while these homologous chromosomes are paired up, you can have a crossing over of genetic material between the non-sister chromatids. So say a little portion of this red, one of the red sister chromatids, can actually swap with a portion of one of the blue sister chromatids. So you can have a swapping of genetic information by a break and repair between two non-sister chromatids. And so you can visualize that here as we undergo metaphase 1, each of the homologous chromosomes are separated from each other, but you can visualize this crossing over shown here. See this little tip that's blue, and over here the little tip that's red. That indicates that there has been a swapping of genetic material between the non-sister chromatids in this situation. And so this increases genetic variability in offspring, because that ultimately will produce a daughter cell that is distinct, is different from the parent cell and from each of the other daughter cells. Okay, so I will just make this a little bit smaller again to show you. During, again, let me back up a little bit. During metaphase 1, the homologous chromosomes are separated into two daughter cells. So you still have the sister chromatids together, okay, but you've got two daughter cells, and then there's a whole nother round of prophase through telophase. This is meiosis 2, where the sister chromatids are separated. So now you've got this sister chromatid being distributed to one daughter cell, the other sister chromatid being distributed to a second daughter cell, and then the same for the offspring of this, of meiosis 1. This will undergo another division, and you've got 4 daughter cells that are each distinct, have distinct genetic information from the parent cell and distinct genetic information from each other, okay? And obviously very important to note is that now you have in these daughter cells, each of the 4 daughter cells, because they underwent 2 rounds of division, but only 1 round of replication, you have half the genetic material, okay? Whereas in the parent cell, you started with 6 chromosomes. Each of the daughter cells only has 3, okay? So for us, again, we have more than 6. We have 46 chromosomes in our somatic cells, but that is divided, and each of our sex cells, the eggs and sperm, will have what we call a haploid number of chromosomes, so half the number of the parent cell. Okay, so these, when fertilization takes place, say this is an egg or a sperm, they will meet with the opposite. And when they join at fertilization, then that restores the diploid number of the parent cell, okay? And then all further growth and development will take place through mitosis, because then that will continue the zygote's proper cell division and growth through mitosis. This process of nuclear division, meiosis, it gives rise to the gametes, okay? So that you have half the number of chromosomes, and then that diploid number is restored at fertilization. Okay, I hope that makes sense. If you have any questions, obviously, as always, don't hesitate to ask, okay? So I've included this next slide just to have a side-by-side comparison of the 2 types of nuclear division that we're talking about this week. So mitosis is on the left, meiosis is on the right. You've got prophase, where you've got the condensing of the chromosomes. Prometaphase is when you've got that breaking up of the nuclear envelope. Metaphase, in mitosis, there is no lining up of our homologous chromosomes. It's just all the chromosomes line up at the metaphase plate and then move to the poles in anaphase and are distributed to the daughter cells in telophase, okay? In meiosis, you can obviously see there's a distinction at the metaphase plate where you've got the homologous chromosomes lining up, okay? And then the first anaphase and telophase that produce the daughter cells of meiosis 1 will still have the sister chromatids together. And then after meiosis 2, where you go through another round of prophase, prometaphase, metaphase, anaphase, and telophase, then you have 4 daughter cells, whereas of course over here you only have 2. 4 daughter cells that are all half the number of chromosomes as you had in the parent cell. Okay? All right, so just side-by-side comparison. And just to verbally point out, beat a dead horse, synapsis is when you have the lining up of those homologous chromosomes. You can have crossing over between the non-sister chromatids, which again increases genetic variability in offspring. Okay, it's a neat technique, I guess you can say, to produce a child that is genetically distinct from either of its parents and either of its siblings. Okay. All right, and then obviously very important is that the daughter cells of meiosis are all haploid. There's 4, they're all haploid. And the daughter cells of mitosis, there's 2, and they're all diploid. And they're genetically identical to the parent cell. Okay. All right. And one of the things we're going to be doing in lab today, besides looking at our slides of nuclear division, we're also going to be exploring the concepts of meiosis in using beads as our model. That is one of the reasons I had made these little bead models, because you're going to be doing something very similar in lab. Now your beads are a little bit cooler. They can connect and be disconnected relatively easily from each other. But since my children at last count had about 70 gajillion beads, I figured I'd take advantage of some of them to show you. You're going to be using beads to simulate first replication of one chromosome, and that will produce its sister chromatid. Okay, now again, you're going to, in meiosis, you're going to demonstrate that these have a homologous pair, so this will be all copied, there'll be 4, all lined up together during meiosis 1, and then you can have that crossing over that increases genetic variability, shown here in beads with the little red bead swapped with the yellow bead. Okay, and then in meiosis 1, the homologous chromosomes are split between the 2 daughter cells, and then in meiosis 2, you've got the sister chromatids divided then between now 4 daughter cells from the 1 parent cell. Okay, so we're going to be working with beads in lab to show each of the stages of meiosis. Okay. All right, so that's basically the gist for this week. You're going to be looking at cells in the onion root tip for our plant cells, and then parascaris for the animal cells. And you're going to be drawing and counting the numbers of cells that you find in each of the different stages of nuclear division by way of measuring how much time the cells spend in each of the different stages of nuclear division. And then we're also, like I said, manipulating the beads to demonstrate the different stages of meiosis and mitosis. Okay, so that is it for the pre-lab PowerPoint for this week. If you have any questions, as always, shoot me an email, cfry@lvc.edu, and I'll see you soon.